We're going to have a look at the rate of photosynthesis today. So this is chapter 4.4 of your Nature of Biology textbook. The key knowledge that you need for this particular one is the factors that affect the rate of photosynthesis. So they are going to ask you about the light availability, the water availability, the temperature, and the carbon dioxide concentration. And many times that these are particularly talked about, it is always in combination with something else. So the factors that affect photosynthesis usually aren't just on their own, they're coupled with something. All right, there are loads of different things that can affect the rate of photosynthesis. One of them is pretty obvious. It is the amount of light reaching their leaves. The next one is the temperature of the environment. We just looked at photorespiration, and of course the temperature is really crucial to whether rubisco will bind to oxygen or whether it will bind to carbon dioxide. So the temperature of the environment is really important for our plants. The availability of water. So if we don't have enough water available, our plants are going to close their stomata. If they close their stomata, they're going to cease photosynthesis because they cannot have the gas exchange occurring. So we need to make sure that our plants have enough water. The concentration of carbon dioxide. So this is a crucial factor because, of course, the carbon dioxide is an input into the photosynthesis equation, and thus we're going to need it. If we don't have enough concentration of carbon dioxide, we're not going to get photosynthesis occurring. Some other factors, the amount of chlorophyll in the leaf. So the amount of chlorophyll in the leaf and the amount of essential nutrients such as magnesium, phosphates, nitrates. Under any of these circumstances, any one of these can become a limiting factor. Now, a factor is referred to as limiting if, in short supply, it restricts the rate of photosynthesis. So I'll say that again, a factor is referred to as limiting if, in short supply, it restricts the rate of photosynthesis. So anything that's going to restrict the rate because of the short supply of that particular thing. So you can see obviously these things here, the amount of light is going to be quite a crucial limiting factor, the temperature, the availability of water and the concentration of carbon dioxide, but also the amount of chlorophyll in the leaf. So a variegated leaf has a lot less chlorophyll than an unvariegated leaf. And the amount of essential nutrients such as magnesium, phosphates, nitrates, all of these are really important in our ability. Like how blood cells need iron, the plant cell needs magnesium, phosphates and nitrates in order to allow photosynthesis to occur. So again, essential nutrients are really important. Sometimes if you look at a plant, and citrus are a prime example of this, their leaf starts to go yellow, and it's a deficiency of magnesium. So that magnesium is needed in the formation of chlorophyll, and of course if we don't have that magnesium there, then our leaf starts to go yellow and we've got no more green chlorophyll, and so thus it can become another limiting factor and prevent photosynthesis from occurring. As with enzymes, our photosynthetic rate increases as the light intensity increases, but it reaches a maximum point. So the graph basically plateaus at the top. On a cloudy day, photosynthesis will not occur at the same rate as it will on a sunny day. Light intensity makes quite a big difference to the rate of photosynthesis. So the ability of the plant to obtain light, obviously that light is then converted into electrons, and through the process it's able to then create our glucose. So without enough light, we haven't got the light dependent phase occurring at its highest efficiency, and therefore we're not going to be able to get our glucose produced at a higher rate. The winter months and the shorter days create quite a limiting factor for plant growth. So we often use artificial lighting, and this also tricks the plants as well. So a lot of the growers of flowers, especially for things like Mother's Day and other particular days, they grow them in glass houses which have plastic around them, so a black plastic around them, and they actually artificially create night and day for the plants. So they create a longer or a shorter day depending on what that flower needs to trigger what that plant needs to trigger the flower's growth. So that some plants need a shorter day to trigger the growth of the flower. So what they actually do is they put the blackout plastic on and they cover it up halfway through the day, and so it, or sometime through the day, and so they create this artificial environment for the plant that it thinks that it's a different time of year, and so it goes, oh, I better start flowering and seeding and reproducing, and so it starts to produce flower buds, and then of course the growers have these beautiful flowers that are growing at a particular time of the year when it's cold, wet and windy outside, and they're in a little glass house and they think that it's in the middle of summer or winter or whatever it happens to be when they flower, and so they can actually artificially grow these plants. This one here, this image here, is a picture of a football field, and they want their football field to look beautiful and green, but of course the problem is that during the winter months we get shorter days. And so the green, overcast and shorter days, and so they haven't got that ability to have a long time of photosynthesis and produce beautiful green lush grass. So we artificially create it by putting lights on them at night time and creating that environment that is much more suited for the plant to grow. At low light intensities, obviously our photosynthetic rate is slow. With increasing illuminance, so as we start to bring the light up, we turn the dimmer switch up basically, the photosynthesis rate starts to increase. The optimal light intensity is the one at which the rate of photosynthesis is the greatest. So we've got our optimal rate here, and of course you can see that's plateauing after we reach that optimal rate. So around here is your optimal rate. And beyond that, further increases in light intensity basically have no effect. So we might be increasing the light intensity a lot here. We're up to this sort of light intensity, but our photosynthetic rate has just plateaued. It can't go any faster. This is what we call the light saturation point. And it means that something else is limiting the rate of photosynthesis. At this point in time, it might be the number of chlorophyll. There's just physically not enough chlorophyll to absorb the light that we're shining at it. So there's some other limiting factor that comes into play here. All right, our wavelengths of light. These are important because we need to understand where chlorophyll is able to utilize the wavelength at its best rate. You can see on this graph we've got chlorophyll A here, and this is the green line. So if we trace this green line, it comes up at a peak here, which matches our blue wavelength of light, comes back down here, wavers along here, so there's really not much happening in the green wavelength. Yellow, again, not really much happening, but then we hit towards the orange-red spectrum, and again we have another peak here. So the chlorophyll A is able to work in orange to red wavelengths and in blue wavelengths. So blue and orange to red is where it absorbs the most energy. Our chlorophyll B, on the other hand, has a big spike here in the sort of turquoise, blue to green area. Comes back down again in the green to yellow spectrum, really useless, but then comes up here in the end of the yellow spectrum. So this is sort of yellow turning to orange spectrum here. It has another peak here. So most plants have chlorophyll A and chlorophyll B in them. They're the two common ones. Plants that contain plants that contain our physoerythrin and phyocyanin, these guys are able to use the green and yellow wavelengths. So they've got photosynthetic pigments here, but most plants have chlorophyll A, B, and that's it. And so some of them will have the carotenoids in them. So our chlorophyll A and B absorb light at slightly different wavelengths, both in the red and the blue spectrums. So this is important to remember. It's a really, it's funny actually, I see greenhouses, and greenhouses typically have, well, used to have, green shade cloth on them. Now have a look here. Green shade cloth is the only thing that plants will not use. So having the green shade cloth on them, if you think about it, if you see green, what colour is being reflected from it? It's green. You see green because green is being reflected from it. So the green shade cloth is absorbing violet, blue, yellow and red wavelengths, but it's reflecting green. Now you stick a plant underneath it and it's reflecting green. What does the plant not use? It doesn't use the green wavelength. So actually the shade cloth that's green is useless for your plants because that green shade cloth is absorbing the blue, violet, yellow and red wavelengths and reflecting the green back down to the plant, which of course it can't use. So shade cloth, you're best off buying a shade cloth that's the fawn colour or a brown colour or a black colour or a white colour. Don't buy green shade cloth. It's not very helpful for your poor little plants growing underneath it. Coming to this, we talk about the wavelengths of light, and my fish tank has a LED light on it now. LED lights are all the rage for fish tanks, and we actually have a look at this fish tank light. It's got, see there's a pinkish glow here and there's a blue glow here. The fish tank lights now are built with red, violet and blue LEDs. If you have a fish tank at home and it's a reasonably new one, lift the lid and have a look and see if you've got red, violet or a blue light in your fish tank. If you have just a fluorescent tube in there, it's probably called a Grolux tube. And that Grolux tube is actually emitting wavelengths of light in the blue, violet and red spectrums. So it's designed specifically with gases in it that will emit a white light, which is mainly made up of blue, violet and red. And it means that those plants that are growing in your fish tank grow really, really well. So there we go. That's why our fish tank lights have our red, our blue, and well, mine is red, blue, and white. So I've got little red LEDs. I've got 2 or 3 red LEDs. I've got 2 or 3 blue LEDs, and then the rest are all white. All right, Engelmann used this experiment here. He had a filament of green algae. So he has this strip of green algae that he put across, and he put it in a tank and he shone different wavelengths of light at this strip of green algae. And he had in the tank an oxygen using bacteria, so a bacteria that consumed oxygen, it fed on oxygen. And he noted that in the tank, the oxygen feeding bacteria, notice they've congregated in the violet area here, a little bit in the blue, not much in the green, not much in the yellow, more in the orange, and then tons in the red area. So you can see that the red and the blue areas here, these were the 2 areas that the bacteria congregated to, which shows you that the majority of photosynthesis is happening in this area here, because what's the byproduct of photosynthesis? That is oxygen. So if this oxygen eating bacteria congregated the most here, this is where the most food is. They would replicate, multiply and divide in this area where there's more food. So you can see just by this little experiment, Engelmann's experiment, it shows that the majority of photosynthesis occurs in the violet and red areas of the spectrum, not in the yellow, green areas here. The ability for the plant to obtain water will also affect the rate of photosynthesis. So we've looked at light intensity and we even broke that down into the separate wavelengths of light intensity. We're now looking at the availability of water. So remember, water is a really crucial input into the light dependent stage. And so if we don't have water, we cannot run the light dependent stage. And so again, this is another limitation of photosynthesis. Not enough water means that we have a problem with photosynthesis. But on the other hand, people don't often realize you can drown a plant. People think, oh yeah, you give a plant water, it's fine, it's just water, keep watering it. But actually plants can drown. You can give them too much water and you then suffocate the roots. So this is why we have holes in pots, so we cannot physically drown our plants. If the soil dries out and the water supply becomes too small, the rate of photosynthesis then declines because we have closed the stomata. We're closing the stomata to stop the water evaporation, But in doing so, closing that stomata also prevents the uptake of carbon dioxide needed in the Kelvin cycle. So we've stopped reducing the amount of water we've reduced the amount of water we're losing, but the problem is we've also stopped the ability for the plant to get carbon dioxide for the Kelvin cycle. So it's a catch-22 for the poor plant. If we have too much water, by waterlogging the soil, the rate of photosynthesis again declines because we've got no oxygen for cellular respiration in the root cells. And so the root cells stop taking up water. And if the root cells stop taking up water, we stop the process of translocation up to the leaves, and we stop transpiration occurring where the water is released from the leaf. So again, we've got another catch-22 where if we give it too much water, we actually drown the roots. The cellular respiration can't occur because there's no oxygen around the root cells, and so therefore we stop uptake of the water. Even though it's drowning in water, we stop that uptake. So it's really important to make sure that we have the right amount of water for our plant. Right, a bit of revision here. The source of water for terrestrial plants is the water content of the soil in which they're growing. So of course that water needs to be taken up by the plant, and of course we've got dissolved nutrients, minerals and things like that in the soil, and of course that's also taken up with the water. So of course it depends on the root system of the plant as to how they're actually taking that up. Water is taken up through the soil in the root hairs. So they're very fine root hairs in the roots. So the roots that you see are the larger roots, but the root hairs are very small. They're almost microscopic, not quite, you can see them with your eyes, but those root hairs actually take up the water and then transport that to the photosynthetic cells via the xylem. Remember, the xylem is the water transport system and only goes one way, whereas the phloem transports all the nutrients in the vascular tissue, and it can go both ways. It can go up or down the plant. So the xylem only goes up, but the phloem can go both ways. Water is then lost from the plants as vapor, and that's your transpiration that occurs in the leaf through the stomata. So it's that capillary action that draws the water up through the plant and then releases that via evaporation in the leaf. So that's our whole transpiration process. When water lost from the leaves by transpiration exceeds the uptake of the water by the root hairs, we have a situation of a water deficit. So if we've got too much water released from the leaf in transpiration and we don't have enough water coming up by the root hairs, we end up with a problem and we've got a water deficit. As our water becomes scarce for the plant, it becomes stressed. And of course, as soon as it becomes stressed, it closes its stomata within minutes to prevent that further water loss. It must maintain as much water in the plant as it possibly can. So by closing that stomata, we've then got an issue. The act of that means that we have no longer got any more gas exchange occurring, and so the uptake of CO2 is interrupted. In turn, this then stops the Kelvin cycle, and so photosynthesis halts, literally just stops, and it alters the balance that normally exists between energy capture and energy use. Remember, the plant has to continue growing. It has to continue using the energy that it's storing in photosynthesis. The whole idea of that plant producing photosynthesis is to power itself. It's not doing it for us, it's powering itself. And so if it ceases photosynthesis, it also ceases cellular respiration, and so that growth can't occur. Prolonged periods of water deficit, therefore, lead to plant death because, of course, the photosynthesis has stopped and the plant can't live if it hasn't produced its own energy source. The waterlogging, on the other hand, creates another issue. So healthy soil supplies water and gaseous oxygen. There is oxygen in the soil, and it transports that oxygen to the root cells and enables that ATP production through the cellular respiration pathway. So the roots need to continue with cellular respiration. If we waterlog the soil, so if we have too much water in the soil and that the soil can't absorb that water, then we have an issue and we've saturated the root zone of that plant. And that in turn causes any air spaces that would normally exist in the soil to become filled up with water. And if that space isn't there, the air isn't there, then the soil, the oxygen content in the soil becomes depleted. Thus, the plant root cells can't respire because they need that oxygen to be able to respire and produce cellular respiration. And so we get a soil condition, an anoxic soil condition, where we have no more oxygen available to the cells. The root cells then begin to die because they can't respire, so cellular respiration is not occurring. And if the root cells die, they're our main cells that are taking up water to the leaves. So if they die, no more water can be taken up by the plant, and therefore the whole plant dies. So even a plant that is watered too much can actually die because of the fact that it just can't get oxygen to the rest of the root cells. And so those root cells die, the water can't get up to photosynthesis, and so photosynthesis ceases and the entire plant dies because it was drowned, basically. And so there we go. The plant will eventually die due to a lack of water molecules for photosynthesis. All right, temperature. Temperature greatly affects how those enzymes and coenzymes within photosynthesis operate. Rubisco, as you know, is one enzyme that is greatly affected by temperature. As we increase that ambient temperature, the rate of photosynthesis also increases. So we have this beautiful incline on the graph. So we have this linear relationship at the start here, where as we increase the rate of photosynthesis with the temperature, we have that beautiful graph. And as a typical enzymatic graph occurs, when we hit that optimum temperature, we have the highest rate of photosynthesis. But again, when we go beyond that optimum temperature, our enzymes suddenly cannot function, they denature, and we get a rapid decrease in the rate of photosynthesis until we get to zero, when nothing occurs and our plant is basically dead. So at low temperatures, remember, we have low collision rates. So we have little kinetic energy, and our collision of the enzymes and the products and the reactants is very slow at that low temperature. If we increase the temperature, then we increase the rate of those collisions occurring, and so our photosynthesis rate will increase as those collisions increase. But of course, at high temperatures, as you know, we then denature those enzymes. And so as soon as we denature the enzymes, boom, our photosynthetic rate decreases rapidly. And we end up with, again, another situation that we don't want to see in our photosynthesis. Our carbon dioxide concentration. As carbon dioxide is one of those inputs to the light independent stage, it's obviously the rate of photosynthesis is going to be affected by the concentration of our input of CO2. It will begin to rise with that linear relationship. So at the start here, as our carbon dioxide concentration increases, so does our rate of photosynthesis in a linear relationship. But of course, as it starts to then get to the top here, we start to have a limiting factor. All right, we have the petering off of the graph, so the leveling off of the graph, and we have limiting factors in here that now, even though we're increasing our carbon dioxide concentration, so know how to read these graphs. They're going to give you a graph like this. Down the bottom here, we have low carbon dioxide to high carbon dioxide over here. On this axis, we have the rate of photosynthesis. So we have a low rate of photosynthesis coming up to a high rate of photosynthesis. Make sure you know how to read these graphs, because they're going to throw one of these at you in the exam, and you need to be able to understand exactly what's going on. They'll have a question related to this graph. You need to understand that you have a consistent rate here. So this is a linear relationship until we hit a limiting factor at this point here. That limiting factor is then no matter how much more carbon dioxide, see how we're still increasing the number of carbon dioxide molecules available, but the rate of photosynthesis remains constant. Look at that, it's still at this number here. It doesn't go above this area here. So our rate of photosynthesis remains constant. At this point here, both the carbon dioxide and the rate are increasing, You can see here, rate increasing. And if it helps to draw the lines on the graph, do it. Do a bit of this. Go, okay, here to here. You can see that the rate and the carbon dioxide are increasing. Look at that. And again, increasing, increasing. And then we get to, my lines really aren't straight, but you get the idea. Look at this. Nothing happens at this point here. And it doesn't matter how much more carbon dioxide we give this graph, we've still got the same rate of photosynthesis occurring. So make sure you know how to read these graphs. They're going to give you a graph like this, and you need to be able to understand that as the carbon dioxide concentration increases, it will increase until it hits a level that it doesn't matter how much more concentration of carbon dioxide you put in here, the rate is still going to be exactly the same. One reason for this limit of the rate of photosynthesis is that maybe the enzymes are fully saturated. So we can't increase that rate because there's only a certain number of enzymes and they're fully saturated with whatever they're supposed to be doing. So therefore, it doesn't matter how much carbon dioxide we're going to put in, we need more enzymes to increase that rate of photosynthesis. So we've got a limiting factor there somewhere. Another factor may be the availability of essential coenzymes like your NADPH from your light-dependent reaction. So if we are limiting one of these coenzymes, again, we're going to have a position where we cannot increase the rate of photosynthesis because we've got a limiting factor. Something is fully saturated and working at its highest capacity, and it doesn't matter how much extra carbon dioxide we put in there, it's not going to get any faster. That rate of photosynthesis is going to stay exactly the same. We've got a lot of interrelationships happening here. So there's various factors that are constantly influencing the rate of photosynthesis. Our carbon dioxide and light are one example of limiting factors that affect each other. They simultaneously affect the rate of photosynthesis. So here we've got the rate of photosynthesis up the side here. Of course, that's increasing as you go up the scale. And you've got your light intensity increasing along the bottom here. So we're sort of turning up the dimmer switch along the bottom here. And we've got two graphs. We've got a blue and a pink. Now the pink one here is 0.03% carbon dioxide concentration. The blue one is 0.13% carbon dioxide concentration. So here you can see that even though at the start here we're increasing at a linear rate, the carbon dioxide is limiting this one here. There's only so much carbon dioxide. Remember, this is an input to photosynthesis. There's only 0.3% available. So yes, it's going to limit the rate of photosynthesis because it doesn't matter how much light you have a limiting factor here of carbon dioxide. So you can see that it's never going to get above this point here unless you add more carbon dioxide, which is what the second graph is. So we've got more carbon dioxide being added here. So we again hit a point where all of a sudden the amount of carbon dioxide, 0.13%, is now a limiting factor. It doesn't matter how much more we turn the dimmer switch up, that light is not going to make any difference because we have fully used as much of the carbon dioxide as we possibly can. And our rate of photosynthesis will not increase above this point unless we A, put in more carbon dioxide, or B, give it more of the enzymes that it needs, which could also be another limiting factor. So at low light intensities, light is the limiting factor. So you can see down here, light is the limiting factor here. So low light intensity, light is the limiting factor. But as soon as these two start to branch, this is where you start to get into carbon dioxide becoming the limiting factor. So here you can see carbon dioxide limits this one to petering off at this point in time. So we get a plateau at this area here. Where this one here, more carbon dioxide concentration, higher plateau. But still, again, we've got that plateau occurring. Temperature is also a limiting factor. Lower temperatures cause lower rates of photosynthesis. If you think about it, the temperature means that there's less collisions occurring. So lower temperature means less collisions. And at this 15-degree range here, we've got less collisions occurring than at the 25-degree range, where photosynthesis is at its optimum. But of course, look at this. We've got 0.2% CO2, 0.2% CO2. So we've kept that factor the same for both. But have a look, it's never going to get beyond this limit here, because now, although we've increased the temperature, we still have a limiting factor of carbon dioxide. So again, carbon dioxide is a limiting factor as well as temperature here, and temperature means that we have a lower temperature, less collisions. So the particles themselves are moving a lot slower, so the collisions are slower. And of course, those collisions are essential for that enzyme to be able to work, because the substrate must fit into the enzyme. So the more activity of the molecules, the better off those enzymes are going to fit into the active site. And that enzyme is, sorry, the substrate is going to fit into the enzyme's active site, and that reaction is going to occur at a quicker rate. All right, I would like you to have a look at your biozone 48 and 49. So 48 is the factors affecting photosynthesis, and 49 is overcoming the limiting factors of photosynthesis. I've set up exercise 4.4 for you to attempt. Many of you are not really attempting these. Have a go at them. They should show you the answers after you've finished, and then go through and analyze what you've got right or wrong. Remember, these exercises are not to be done as a test. You can use your notes to help you answer the questions. These are the questions out of the back of the book. So these are the questions for each chapter, as well as exam questions. I want you to have a look through your notes as you're answering these, so you can effectively answer them. You don't have to answer them like blind, as I would say. Go through your notes and help you answer the questions to get you a better understanding of what's going on. Then have a look at Enrollo. Enrollo for this particular unit is brilliant. 5C, factors affecting the rate of photosynthesis. This is a really good one to have a look at, because again, he goes through all of these details in there.